We're now just about the end of the first day of our 2-day IEDB virtual user workshop. Before I move on, I just wanted to say a huge thank you to all of our presenters for today. You've been fantastic. The virtual space can always go a little bit awry. So yes, I thank you so much for all of your patience. To the attendees, I know you're up at all sorts of hours. It could be day, it could be night for you as well. I know a few people had to drop off because it was well into the night for them. And we're just really appreciative that we have such a keen user base that wants to stay on and learn more about the IEDB and the other NIH resources. So just to quickly move through, as I mentioned, one of our key goals from the start of this day, we talked about improving our resources through your user feedback. And that not only means the IEDB and analysis resource, but also the events that we have like the user workshop. So in the chat, I have shared the link. As I said, please do fill it out for today, and we will take this forward and look at improving our event again for next year, which may be virtual again. We might be back to in person, who knows at this point. So yes, that is the link. We'll also be sharing another link for tomorrow for day 2 feedback, and then a third survey well into next week with a follow-up email just to get your feels for the 2 days overall. So as I've mentioned a couple of times, please do take some time now to fill out the survey. We tend to forget if we leave it until after the event. So if you could start to fill it out as I'm concluding, that would be fantastic. And I briefly just want to give you a recap of day 1. So we kicked off the day with Alex giving us a database overview, where he talked about the main search interface and some of the results and the key features around the querying of the database. Next, we had Randy join us on how data is retrieved, entered, organised, and accessing the data. So a wonderful in-depth presentation with some key examples, answering some key user questions that were resubmitted, and showing you some more detailed examples on how the search interface can be played with, some of the finders, and as I've just mentioned, the queries, and an in-depth look at some of the other features like the immunome browser. Then we had Betty and Will join us from the Los Alamos team to present on their COVID pipeline work. And then right towards the end, now we've just had Bjorn give us an overview of the analysis resource in particular, overall rather, and then Alex just joined for the analysis tools in particular. So the population coverage, cluster tool, and so forth. And so today was really just a broad strokes overview of the analysis resource. Tomorrow, we really will get into the nitty gritty detail with some of the other bioinformatics team members. So if you're bioinformatics inclined, tomorrow will certainly be your day. And then lastly, of course, we had Alba presenting on how she and the others in the team were able to use the IEDB database and analysis resource in their SARS-CoV-2 research. And that's just a few of the different aspects that Alba used with the immunome browser, the database Discotope tool, and the 3D viewer. So the workshop structure, day 1. Day 1 is complete now. Again, thank you so much for your time. As I mentioned, tomorrow, day 2, we will be starting at the same time, 8 a.m. Pacific time, and it will take a really strong look into the analysis resource, the T-cell prediction tools, B-cell as well. So we'll really break that down and hear from the other team members that you didn't get to hear from today. And then just a quick preview of day 2. So we will start just shortly after 8 a.m. PT, where Bjorn will go into the MHC binding predictions. And then we'll have a short Q&A where Jason Greenbaum will also be joining Bjorn there. After a brief break, Bjorn will come back with our research technician, Austin, and they will present the T-cell processing and immunogenicity predictions. So that's a great part. And then Will will come to the fore, and he's presenting a new tool that is not available to external users at the moment, TCR Match. So some things we're really excited about in this user workshop. We're giving you a behind the scenes look on some of the tools and ideas that we have in progress. you've definitely heard from a few team members that, we're planning to do this, or this is in motion. And so we have an opportunity here for you to see the TCR Match tool in a live demo as well. So we'll be taking us through that. And then we'll be joined by Paolo to give us a look at the structure tools, Lyra and Scepter. Another Q&A, and then a bit of a longer break, that 30 minutes. Then we'll have Brian and Elizabeth Sharon Fung from Los Alamos joining us to present the other half of the LANL presentations, which is the HIV databases. They'll also answer a few questions. Then Bjorn will present on the B-cell epitope prediction tools. And then Jason Greenbaum, will be presenting the future of tools. So the IEDB Tools 3.0, this is another really exciting part that we get to show you a behind the scenes view of something that's in the works. It won't be available for a little while, but you're getting a first look at things that we have in motion to make your user experience even better. And then we'll wrap up the same with a few closing remarks. And that brings us to the optional Q&A portion of the session. So again, I would really like to thank you all, to all of our attendees, for joining us today. I know, 6 hours is a long time to be sitting at your desk. It's obviously a lot more fun when you're in person and interacting with all of your colleagues. But no, we really appreciate your time, and we hope you learned something new on this day one of our 2-day event. Our team will be sticking around. So there are no new questions in the chat box as of yet, but we'll stick around for at least another 15 to 30 minutes, depending on how many questions come through. And they'll be around to have a bit of a chat and answer a few more questions if you would like. So with that, I would like to thank our presenters, thank all of our attendees once again, and I will leave this here in case there are any few questions, and I see there is one that's just come through. So Alex or Bjorn, I think you might want to take this one.
So this falls in the category of the TCR tools, and tomorrow we're going to present the first one that we have. We don't have one that specifically does this. Actually, we have development ongoing to do exactly this, but we aren't there yet.
And maybe just repeat the question, sorry.
Okay, sorry. The question is there a tool to predict HLA restriction for TCR CDR3 sequence? My suspicion is going to be, it's not going to be CDR3, but it's going to be CDR1 and 2, which have the contact with the MHC molecule, versus CDR3 is primarily with the epitope itself, which makes it most interesting for identifying specificity. I would expect that the tool is going to be better to do so in principle, it should be possible to do something like this. And I hope I answered this. So I guess the quick answer again is, like, we don't have one currently. I'm not aware of one that does this systematically and globally. But it should be possible in principle, and I doubt it's going to be CDR3, but I expect it to be CDR1 and 2.
Excellent, thanks, Bjorn. There are no other new questions as of yet, but here we go. As soon as I say that, just give the team a few moments to have a look.
This is one for Alex, I think.
The question is about T-cell activity recognition assays. From previous experience in a wet lab on tumor immunology, I grasp the idea, but depending on how interferon gamma release are performed, the degree of positivity can differ, making types of interferon gamma assay difficult to compare. Is this taken into account when curating the assay data? Do you share this view? So there are several answers to this question. Wonderful question. So one thing is that indeed different interferon gamma assays are not necessarily comparable. And in fact, this goes back to discussions we had early on, where for example, we did not put ELISA titers when people were running interferon gamma ELISAs, because a titer of 1,000 doesn't mean anything, because depending on what antibody you use, what conditions, it creates a false feeling of quantitation that then could be really messy if you're trying to compare different assays. So certainly there are interferon gamma assays based on whole blood cell assays, ELISA assays, ELISpot assays, and so forth. And those are not comparable. More in general, I think that there is a growing movement towards using agnostic assays, because basically if you're using an interferon gamma assay and the particular T cell you're studying, T cell response you're studying doesn't feel like making interferon gamma, but is instead making IL-2 and IL-10 or IL-5, you'll be blind. So there are activation-induced marker assays now that are routinely used, where you just basically detect what kind of activation occurs without necessarily measuring a particular functional outcome. And I can take also the next one. The immunobrowser is similar as the hotspot analysis for MHC elution data. Yes, it is. And in fact, that was one of the motivations why we decided to also make the immunobrowser available as a standalone tool, because people routinely generates for class 2 this elution data, which is very difficult to visualize, because the class 2 eluded peptides have ragged ends, and they are many different things that are overlapping. And so that is ideally, it's an ideal application to show the immunobrowser, because it does visualize the regions across a sequence. And in this, you're probably familiar with the MAP type of assay, MAPP.
Okay, I'll take the next one, which I'm not qualified for. Where do you recommend to take a Master of Bioinformatics? Very interested in the creation of databases. So the reason why I'm not qualified for it, we are not a teaching institution, and I'm not familiar with enough programs to make strong recommendations. If you're specifically interested in immunoinformatics, it has to be in Europe. I would very much recommend going to Copenhagen. We've gotten fantastic people coming over from there for us to visit, and they have probably the best program that I'm aware of, specifically for immunoinformatics if you're located in the US. And obviously, we think UCSD is a great school, and they have a very broad program, and you can pretty much do anything you want. There's a lot of bioinformatics programs out there, and those were my two personal biases. And I'm sure there's going to be more. I'm not sure if the audience here is necessarily the best. Us speakers here necessarily can judge all master bioinformatics.
So the next question is, there have been reports comparing binding strength and immunogenicity, which report that high binding affinity is not always related to an immune response. What would be the recommendation for immunogenicity prediction data results from IEDB and this fact? Yes, that is true. And we have been very emphatic in saying that the fact that something binds does not mean that it will be immunogenic. HLA binding is a necessary condition for something to be immunogenic, but it's not sufficient. And the same applies to naturally processed peptides. So to a certain extent, something needs to be generated by natural process in the course of an infection or an immunization in order to be recognized. That also is provided that you have enough sensitivity to detect, because T cells can often get activated even by a single copy of a peptide. But the truth of the matter is that that's something also Bjorn pointed out in his introduction, that the predictions, whether they are predictions for binding, for eluded peptides, or whatever, need to be experimentally verified. And again, also, whether something that binds is going to generate an immune response is context dependent. That has to be understood, because if you have a peptide and you inject this peptide in mice in saline, nothing will happen in most of the cases. Because the peptide will be instantaneously degraded by serum proteases, or B, in fact, in the old times, that was a methodology commonly described to induce tolerance, to induce the lack of a response. If you inject the same peptide in the context of a strong adjuvant, you might induce a response. So there's exactly the same peptide that binds exactly the same binding affinity. So again, binding affinity is a condition that is necessary but not sufficient. Furthermore, we have been working, and Bjorn has been working on this topic quite a bit. You can actually derive functions that predict the likelihood of something being immunogenic based on a combination, almost like a Boltzmann type of equation, combination of the binding affinity and actually how much of the peptide is available in terms of mRNA expression and so forth. So to a certain extent, this can be refined in a quantitative fashion, but predictive binding is not a free ticket for guaranteed immunogenicity.
Okay, so I have a question which says, I see in the general IEDB metric, T-cell assays are behind B-cell assays. The community effort to treat this source of data. Second, maybe asked before, but when a T-cell epitope is considered immunogenic, immunogenicity equal yes, does this imply that it has been tested against all the MHC alleles included in the MHC restriction? So with the first question, yes, currently we have, I think, like 100,000 or 500,000 B-cell assays and 300,000 T-cell assays. These have been driven. These have been driven by a few experiments that were going after a large volume of assay data. So there has been like a peptide tiling microarray that gave like 100,000 new assay data points. So I would not say that T cells are generally behind B cells. I hear a comment here, I'm breaking up. Is that for everyone?
Yes.
Is that just started?
It was happening while you were repeating the question. You're a little bit better now.
Oh, that's weird. Okay, so but I was first trying to say, actually, I think currently there's like, it's got a back and forth between T cell and B cell assays, and there's large papers coming in reporting hundreds of thousands of data points that we would consider separate assays that are kind of changing the balance there. So I don't think there's definitely a need for specifically that T cells are lagging. The other question was, so immunogenicity for us means that there is a T cell response that was detected. And if at the same point, that might be done in this worst case in a single individual. And at the same time, the same peptide might be tested against 10 different MHC alleles for binding. And it might be then reported as being immunogenic, binding to all these alleles. So in that case, that would not imply that all that binding is always also tested for immunogenicity. You will have to go into the specific data points. And so there's no implication here that both is always done symmetrically.
So I don't know if our Los Alamos friends are still with us. Anybody?
I think, Will, if you're still on the line, it looks like there's one question directed to your presentation with Betty. Maybe we can take that one offline.
Right, maybe take that offline. There is a question whether the IEDB includes data and tools for peptide MHC binding stability. It does not. When we looked at the data, so as a context here, there has been reports that implies that peptide stability is a better predictor of immunogenicity of peptide binding. It's true, maybe, but really the difference in our read was rather minimal. And so that had to be balanced to the fact that the data available to train algorithm based on stability data, it's rather minimal. So considering those two factors, rather limited increase in performance and generalized lack of training data, that is why the answer is no.
I think, Nina, we should stop it here. Seems to be a good end.
I think so too. So yes, we have finished a little bit early, but I think we've got through all of the questions, which is quite a feat. I can see we've answered over 60. So that's amazing for day one. And I think I will leave it there. Thank you, Alex, Bjorn, for answering those few additional questions. This last question I will pass on to the LANL team, and they can take that offline for you, Guillermo. And with that, I would like to thank you all once again for your time. And we hope you have a lovely day, afternoon, evening, night, and hopefully see you again tomorrow, 8 a.m. Pacific time.